Hello, everybody. I am going to be going through the lecture today, just as a reminder that we're not having class tonight. And then we will be back on our normal schedule next week. So here is the lecture for tonight. Let me just pull it up. Sometimes it doesn't want to. Let me just go back really quick. Sorry. Okay, so we are doing mutations and variations, which goes along with evolution. A couple people emailed that the evolution videos are not working. I will try to get in there and fix those today. I'm not sure that I can find the original, so I might have to come up with a few of my own, but I just wanted to let you know that that is a problem. All right, so the essential concepts that we're covering today is that basically the natural selection piece on how all of these mutations that we've been talking about and transcription translation comes down to mutations that give a rise to new phenotypes or alleles, and then how it is favorable versus unfavorable to fewer offspring. So here we have variation that is found in all populations. This is the seed cracker. This is an American African genesis of a bird. It has a lot of different variations and beak variations, but the point that we're looking at is that even though the beaks are different, the structures within the bone are actually very similar. And so the beak might be different, but the bone structure for the bird itself inside that as the base for that beak is very, very similar to each other. So there is just a slight variation on how that beak is presented, but overall, it doesn't change the bird itself and its structural piece. Selection sorts out variation. So this is an example of how this different antibiotic piece, the different resistance happen first based on selection. So you have a bacteria that if we're looking at the left one, it is showing those clear rings. That means that no new bacteria is being created. So that antibiotic sensitive piece is working. So the antibiotics are working on those particular bacteria. But if you look at the right, the antibiotics pieces are not working anymore, and the bacteria itself is now resistant. So the way that this works is we have this before selection. So what this is based on is that the bacteria itself at one point has a variation of what's resistant and what's not. And so when we include an antibiotic, it carries out and clears out all of the resistant or the lack of resistance bacteria. So it kills all the ones that have no resistance whatsoever. And then we're left behind the ones that are more resistant. And so this is how this process is working. So we're basically, if you look at the down here at the bottom, the low resistant bacteria are the lighter ones. And as we kill those off with the antibiotics that we use, after that happens, we get more and more of the antibiotic resistant bacteria. And so then it is left behind higher populations of those very resistant levels of bacteria. So this is kind of how the selection piece works as far as variation and how it starts using the example of antibiotic resistance. So variation is a result. One of the key pieces are that variation is a result of mutations. That's where most of our variation comes from, is the mutation of those genetic codes and how they're connected. But it gives away to new alleles. So you have these alleles that are gene variants. For example, the insulin growth factor for dogs. There are often multiple alleles for each gene in the population. However, if we're looking at this one right here at the bottom, you have this one gene that is different from large to small dogs. So you'll see these small dog, this gene variation is usually in smaller dogs like Chihuahuas, and that kind of thing. So think about this, what kind of mutation is this? It is a point mutation, yes. And we would have to go through the process of finding amino acid to see which one it is, if it is actually a missense, a nonsense, um, or a silent. So we'd have to go through and figure out the mRNA that matches that to figure out which one it is. So if we're looking at mutations overall, they can be three things. They can be neutral, they can be harmful, and they can be beneficial to an organism. So they arise in a genome randomly, so they don't always show up normally, obviously, they're random. But how they affect the organism's fitness depends upon where and how they alter the genome. So we already kind of talked about this a little bit with our case study in class. But let's just go through this. So this is one of the most influential genetics scientists that has ever come to play. He proposed that most mutations are neutral, and genetic drift is the critical point of evolution. So if we're looking at, for example, the mutation is part of the genome that isn't protein coded or regulatory. So those are the two options that he gave. As far as how these mutations usually occur. And then example two would be a silent mutation in the protein coding region. So he again said that there are basically more neutral mutations than there are that are harmful or beneficial. But it is also both based on the genetic piece. So if we're looking at mutations that are sometimes harmful, this is one of those cases. So, Layman A gene causes progeria, which is a premature aging resulting in early death. So, this is a missense mutation, which hopefully you remember is when the amino acids are changed. They're not the same, they are basically different amino acids. And this is harmful not only because most of the people that have this don't get past the age of 13, but it also tells us that there's no reproduction. So that actually is, besides the early death, no reproduction happens in those people that have this or the patients that have this. There's, like I said, they usually don't live past the age of 13, and so no reproduction is included in that. And that's one of the things that is very harmful to a species in general. Okay, so I'm going to have you think about this for a second. Stop the video, look at this again. What type of mutation is this? So here are the patient, here's the normal. So this is the sequence that we're looking at. Here's the normal sequence. Think about this for a second. Stop the video. I'm going to give you a second to think about this. Stop the video and see if you can figure out which one of these cases it refers to. So hopefully, oops. So hopefully you realize that this is a point missense mutation. The point is that there are just a one amino acid nucleotide here. This first one, the C. Now, again, these are the DNA structure, but remember we'd have to transfer this over to RNA. But T and C obviously would end up being different nucleotides even in RNA. And so we get two different amino acids, which is our missense. So it's this guy right here. It's a point because it's only one piece, one nucleotide, but it's a missense because it's giving us a different amino acid. So hopefully that got you the right answer and that you figured that one out. Going on to mutations sometimes being beneficial, we have this deletion in the CCRF5 gene. People who inherit this don't express functional proteins on the surface, and so they are more resistant to things like HIV. So another piece of this that is beneficial for people who are not affected with HIV is that it can sometimes protect against things like smallpox, bubonic plague. I know that sounds like archaic since we don't have very many conditions like that, but that's just the beginning. They think there might be other types of conditions that this helps people be more resistant to. So it's not it's archaic because it's smallpox. We haven't had smallpox in whatever, it's eradicated. But we all know that things like smallpox and bubonic plague, even though we haven't had conditions of it, there's always a chance. But there's always a chance they think that this might help with other resistant pieces to other conditions other than smallpox and bubonic and HIV, that there are other conditions that this may be a beneficial tool to keep someone resistant to certain diseases. So one of the first geneticists to use DNA to trace African ancestry, which is an important piece. It's important to note that when we're looking at genetics, race is not a thing for this. It's race versus ancestry. Race is not a biological thing. It's a social tag that they include in this. When we're talking about anyone with a different descent, we look at ancestry. So ancestry is genetics and it's a biological piece versus just race. So just keep that in mind. But he did a study on African ancestry and studied genetic markers to estimate the risk of diseases and responses to medications for people of color or genetics of skin color. And then he published health disparities on this as well. So he looked at the genetics of skin color and how many were involved, how many genes are involved. And there are about 100 loci that have the exact number. We don't know for sure how many actual loci or actually affect what the skin color is. But it also refers to pigmentation. So natural populations or animal models are included in this. So beneficial pieces. One example is their Komodo bear. So there's a black bear population in British Columbia. Most of them are black. That's what they are. But then 10 to 20% of that population has light fur. So they're also called spirit bears by the First Nations. And so with that being said, the study for one of his was not only just the skin color of people, but also the variation of color in different species. And this was one of the studies that he did or helped with. And so one thing to note that the Komodo bears albino, no, they are not. And the reason that they're not albino is because their eyes and skin are still pigmented. So that is not what these are. So they're not albino, so they still have those pigments in other areas. It's just their fur that is the issue or not issues. So let's think about this for a second. Think about this in your head. Pause the video. How could you determine genetic basis for light fur in Komodo bears versus darker fur in Komodo bears? So I'm going to give you a second to figure that out. Pause the video, think about it, come up with an answer, go. Okay, hopefully you have stopped the video and you've thought about this a little bit. So when we're looking at a candidate for gene approach. So this would be this particular piece. The sequent genes are known to be involved in pigmentation. So the ones that they're studying at, look at that and look for pigmentation. So if we're looking for the difference between dark and light bears, this is the study that came up. And so they looked at like 5 different types of candidate genes to determine which one of these would be the outlier or the one that causes it. And albinism, we already have determined that it is not one because of the pigments left in the eye and other areas of the body, not just the fur. So they went through and decided that the melanocortin 1 receptor is probably the mutation piece. So if you look at this sequence, and hopefully this looks familiar to you now, but this is not mRNA, this is DNA. So in order to figure out what type of mutation it actually is, as far as missense, nonsense, or silent, you would have to transfer it over to mRNA. But for DNA, just these pieces, you can look at it and say, oh, it is what kind of mutation. So we have insertion, and remember, insertion is where things are inserted in, so it frame shifts everything. Then you have deletion, which takes one out, so one of them would be different and would be gone. And then point mutation, which is just an exchange of a nucleotide in that place. And so if you're looking through this, and remember, we're still reading it, we still have to have 3 codons per each particular piece. You would have, for instance, if this was a deletion, you would have one less on this strand as far as what they would be looking at. And if we had an insertion, you would have one more added to the strand. And so since we don't have these, hopefully you came up with the point that this is a point mutation. So in other words, it's just exchanging the A on the dark bears for the G on the light bears. And the codons are a little bit different as well. So if you went through this, it may not look like anything, but there is that sequence difference. And so hopefully if we were to exchange this for mRNA, there might be an exchange of the amino acid as well. So if we're looking at this point mutation, if you're looking at it, this is an exchange of an amino acid. So this is usually it's trypsin, and it goes to cysteine. So there's an interchange here. So what kind of point mutation is this? If it's exchanging a amino acid for a different one. So think about that for a second. Go. All right, hopefully you have chosen that this is a missense. It's actually calls for a different amino acid in this particular strand of DNA when it's actually translated. So that is a problem as far as keeping it the same. So the dark bears and the light bears in this specific area of their amino acid chain has a different amino. So what this actually does is this is basically a receptor. There's a receptor for this particular hormone, this protein that's coming in. And so that receptor actually regulates the dark and the brown colors and the lighter colors of these pigments. And so what would happen if this mutated so that it had a higher binding affinity for an MSH change? So keep that in mind. Think about it for a second. What would happen? You predict it, right? Transcripture of genes. So in other words, if we're looking at that receptor, we're looking at different animals here or species here, that this receptor will cause different color variants for species. And so a few of them are shown in here. You'd have to kind of go through it a little bit. But going back to this one, hopefully you chose that it would higher binding affinity, which means that it would stay there longer, which means that receptor would not actually transcribe as much. So there should be a decrease in the production of these. So then you get this result. You get the light colored bear, you get fair colored skin. This is specifically for redheads. This is one of the receptors that causes this lovely color to show up. And then, of course, there's variations here, which where those receptors are basically stopping or increasing some of those pieces to have a different color variance in different species. And these are just some examples of those lovely little guys at the end. So again, the color code is called polymorphism in mammoths, which is one of the things they looked at, and it works the same way. It has that silent receptor versus the non-silent receptor, depending on which lovely amino acid is included into that. And then you get things like different colored fish, which is the things that you were doing for your assignment today. Okay. Okay, so we've gone over the lecture. I wanted to give just a quick, very quick idea of what you're doing for your assignment. You guys should have picked these up on Monday. If you did not, they'll be attached to the in-class assignment area. Remember that these in-class assignments are still due by Friday or Saturday night, although I will extend it through Tuesday if you need additional help or need some clarification. So we're going to go through really quick the assignment that you guys are doing tonight or today. So this is what the assignment looks like. So you're looking at genes that include different color variations that are showing up. So sorry. So the purpose is to let you analyze mutations in a gene sequence, the gene function, and how it affects the fitness of the species or the relatedness of the organism. So you're going to be using interpretation of data, and then you're going to classify mutations as we go. And we're looking at the melanophores of the zebrafish skin. So there's a possible role of the genes in human skin suggested by a group of researchers. And they're looking at how those genes with mutations might lead to cancer. But one of the variations is analyzing how is known as the golden mutant. So basically a mutant compared to the wildlife fish. So we're basically using that same different variation of gene in two types of fish. So we have the wild type zebrafish versus the golden mutant fish. And you're basically going to read through these particular fish and what their descriptions are. And then you're looking or answering a few questions. For the golden allele, you're mapping out the chromosomes in those fish. And there's two alleles that are resulted in that golden phenotype, the GOL B1 and the GOL B13. The region contains the gene that we were talking about before, and there's markers on each. So you're looking through the data and then you're going through the golden alleles, and then here is the gel below. So you basically did a PCR result or like a gel electrophoresis result. And the bands indicate that DNA is present, and if there's no band, it means that that region of the DNA is not available or not there. So you're going to look through and hypothetically look at the results and then answer a few questions. And if you're not sure, here is the wild type strand versus the mutant strand. And then you can kind of decide which kind of mutation is involved in this color change between the two fish and what it might look like as far as injecting the molecule and targeting that gene on each one of the fish that we're looking at. So the golden and the wild type. And then you're just looking through a number of vertebrates and looking through the alleles and comparing them to different ones and then answering just a few more questions on how they're different or the same or what the variations are, what's missing for each one of those. And it's kind of an interesting little study, a little bit fun kind of going through the differences between each one of those species. All right, I know it's a lot of information. Actually, it's actually kind of a fun one this time. I wish we were doing this kind of in person, but due to the fact that, there's a lot of scheduling issues at Slick this today because of the 4th of July tomorrow. But I just wanted to go through this with you. Again, your normal schedule applies. It should be done by Saturday night, but I will leave it open until Tuesday. Do not do that though, only if you need questions answered. And then hopefully we can get back on track next week with a normal schedule. So again, if you have questions, contact me through Slick. I hope you guys have a wonderful 4th of July, and I will see you on Tuesday.